This video presentation aims to illustrate drug release and absorption mechanisms with the help of mathematical modeling approach. The continuous demand for designing more reliable delivery systems requires the use of the most updated theoretical tools. The old trial and error approach is accepted neither by academia nor by industry anymore, whereas new approaches reduces product development time and costs, and it also minimizes the risk of unsuccessful investments. This strategy adapts well with industrial demand for simple and reliable tools aimed at improving product characteristics and at exploring new market regions. Drugs are introduced into the body by several routes. They may be taken by mouth, given by injection into a vein, into a muscle, into the space around the spinal cord, or beneath the skin. It can be placed under the tongue or between the gums and cheek. It can be inserted in the rectum or in the vagina. It can be placed in the eye or the ear. It can be sprayed into the nose and absorbed through the nasal membranes. It can be breathed into the lungs, usually through the mouth and nose, or applied to the skin cutaneously for a local or body-wide systemic effect. Each route has a specific purpose, advantages, and disadvantages. Anatomically, the stomach can be divided into three major regions: fundus, corpus, and the antrum. The main function of the stomach is to process and transport food. After eating, the contractile activity of the stomach helps to mix, grind, and eventually evacuate small portions of chyme into the small bowel, while the rest of chyme is mixed and ground. Four major types of secretory epithelial cells cover the surface of the stomach and extend down into the gastric pits and glands. First, mucus cells which secrete an alkaline mucus. These cells secrete a bicarbonate-rich mucus that coats and lubricates the gastric surface and serves an important role in protecting the epithelium from acid and other chemicals. Second, parietal cells which secrete hydrochloric acid into the lumen, where it establishes an extremely acidic environment. This acid is important for the conversion/activation of pepsinogen to pepsin and inactivation of ingested microorganisms such as bacteria. Third are chief cells which secrete pepsinogens and inactive zymogen, which once secreted is activated by stomach acid into the active protease pepsin, which is largely responsible for the ability of the stomach to initiate digestion of proteins. And fourth are G cells, which secrete the principal gastric hormone gastrin, a peptide that is important into the control of acid secretion and gastric motility. The stomach absorbs very few substances, although small amounts of certain lipid-soluble compounds can be taken up, including aspirin and other non-steroidal anti-inflammatory drugs and ethanol. The intestinal mucosa is a selective permeability barrier routes which can cross drugs through the transcellular and paracellular routes. In the transcellular transport process, the drug molecules have to go through the barrier cells, the epithelial cells, to reach the systemic circulation. It's a two-step process, starting with the drug uptake into the cells and ending with the drug efflux out of the cells. The second type is called the paracellular transport process, where the drug molecules travel between the cells or in the gaps to reach the systemic circulation. It is possible that both routes contribute to the absorption of a particular drug. However, in many pathological conditions, the integrity of the intestine wall can be altered to different degrees and abnormal absorption properties can occur in the diseased intestines such as Crohn's disease, celiac disease, food allergy, and pancreatitis. The human skin represents the largest organ of the human body, which is composed of three layers which differ with respect to the anatomical localization, structure, properties, and embryological origin. Drug application to the skin can be devoted to treat dermatological disorders called topical deliveries, treat deep tissues such as muscle and vein called regional delivery, and to allow the penetration of drugs to the systemic circulation as transdermal delivery. The goal of the topical delivery is to create a reservoir of drug in the skin. Penetration of the active molecule to the deeper skin layers or systemic circulation is not necessary. Regional delivery requires deeper penetration than topical delivery, as the drug has to reach muscles and veins. Transdermal delivery aims to treat systemic conditions by penetration of drug into the systemic circulation. Topical delivery substantially does not require to be improved to achieve therapeutic dosage of the drug at the disease site. Regional and transdermal delivery needs to be implemented by enhancement methods able to interfere with barriers such as electrically assisted enhancement, vesicular carriers, chemical enhancement, microinvasive delivery, and velocity-based techniques. Rheology can play an essential role in the study and development of drug delivery systems, not only for solving microscopic problems connected with their preparation and application, but also to examine the structural features and processes that govern their behavior in flow conditions as well as under small deformation. The classification of the rheological behaviors is not a simple task, because every material can behave differently under different conditions. Changes in applied stress or deformation conditions induce structural changes whose amplitude and kinetics depend on their intensity and time of evolution. All these considerations are relevant for the pharmaceutical sector, where the majority of the materials like ointments, creams, pastes, and gels are viscoelastic, since they combine both viscous and elastic properties. Rheology can not only improve efficiency in processing but can also help formulators and end users in finding pharmaceutical products that are optimal for their individual needs. Pharmaceutical processes such as ingredient selection, preparations, material packaging, and shelf storage are often associated with complex flow conditions of materials. The application performance and hence the acceptance of the final product are also often dependent on its rheological behavior. In order to exert a therapeutic effect, a drug should have a high affinity and selectivity for its intended biological target. For example, a protein or a protein complex in or on particular cell type, and also reach a sufficient concentration at that site. The drug has to be released from the delivery system, transported from one side of the application to the site of action, biotransformed, and finally eliminated by metabolism from the body. Thus, the whole process can be divided into the pharmaceutical phase, where drug liberation takes place, and the pharmacokinetic phase, describing the course of the drug in the body and its metabolism. Mass transport is very important also in the pharmacokinetic phase, as compounds are absorbed in the body on condition that they cross cellular membranes and takes place according to passive diffusion or to active mechanisms. Although metabolism and physiological factors highly affect drug absorption by living tissues, bioavailability strongly depends on drug permeability through cell membranes and drug solubilization in physiological fluids. If solubilization is the first absorption step, permeation is the second one, as drugs must dissolve in the physiological fluids and then it must cross cellular membranes. Solubilization implies the drug dissolution process, and permeation implies drug partitioning between a polar aqueous phase and a non-polar phase like cellular membranes, unless active mechanisms rule drug permeation. Examples of commonly marketed drugs that are poorly soluble in water that is less than 100 milligrams per centimeter cube include analgesics, cardiovasculars, hormones, antivirals, immune suppressants, and antibiotics. Drugs with improved water solubility can be administered in a lower concentrated dose, with a reduction of local and systemic side effects. This is crucial for drugs with important side effects, such as antibiotics, antifungals, or antivirals. The importance of both dissolution properties and permeability on drug delivery systems is very well emphasized by the biopharmaceutical classification system, setting the regulatory framework for drugs classification. The first step consists of the contact of the solvent with a solid surface, wetting. which leads to the production of a solid-liquid interface starting from solid to vapor. The breakdown of the molecular bonds of the solid called fusion and passage of molecules to the solid-liquid interface called solvation are the second and third steps respectively. The final step implies the transfer of the solvated molecules from the interfacial region into the bulk solution, which is the diffusion. A detailed study of drug partitioning between a polar aqueous phase and a nonpolar one is very important since some drug physical-chemical properties and in vivo behavior can be determined on the basis of this. In particular, the drug partition coefficient P has an importance in predictive environmental studies as it is used for the prediction of distribution among environmental compartments, for the estimation of bioaccumulation in animals and plants, and in predicting the toxic effects of a substance. According to their release behavior, controlled release systems (CSRs) can be classified into passively pre-programmed, actively pre-programmed, and actively self-programmed systems. While 15 years ago the majority of CSR fell into the first category, at present the importance of the last two categories has considerably increased. Only the last two categories can fully accomplish the modern concept of therapeutic treatment, which is to increase drug effectiveness and patient compliance. Controlled release matrix systems, defined as three-dimensional network containing the drug and other substances such as solvents and excipients. Matrices can be prepared by mixing the drug in the form of a thin powder with a pre-polymer. Then, the whole mixture is poured into the polymerization reactor. Polymer science, surface, bioadhesion and thrombogenic properties, drug matrix interactions, drug physico-chemical and therapeutic characteristics, and biocompatibility merge together to determine the final reliability and effectiveness of a matrix-based delivery system. One of the most common approaches to get controlled release is to embed a drug in a hydrophobic or hydrophilic matrix to speed up or to reduce drug release kinetics depending on the final therapeutic target. Matrix-based delivery systems find a great variety of applications in the pharmaceutical field, especially in oral administration. In regenerative medicine, matrices like hydrogels are used as three-dimensional support for growth of cells, as in the case of stem cells isolated from the human bone marrow and chondrocytes. Hydrogels are also suitable to release proteins and oligonucleotides, high molecular weight and fragile drugs, which can easily undergo denaturation or degradation during the formulative process. Microemulsions are powerful drug delivery systems as they can be both suitable for the release of both scarcely water-soluble drugs and enzyme-labeled water-soluble drugs. In addition, ease of fabrication, versatility of administration route, and low production costs make them very attractive for the delivery field. Nevertheless, microemulsions are complex systems whose structure can be modified by external conditions jointly with the presence of the loaded drug. Drug diffusion through membranes is an important phenomenon both for synthetic and biological membranes. Indeed, synthetic membranes can be used to measure drug diffusion coefficient, and this turns out to be very important in all the delivery systems based on membrane-controlled release kinetics. This is the case, for example, of microencapsulated systems where synthetic membrane permeability is one of the most important parameters for ruling release kinetics. At the same time, drug diffusion in biological membranes, specifically intestinal mucosa and skin, is of importance for the wide practical application of oral and transdermal administration routes. In addition, drug permeability is one of the most important drug properties together with solubility for the determination of drug bioavailability.